Shai Arkin. Professor Shai Arkin is Vice President in the University and the Head of the Authority for Research and Development. And besides being Vice President, he is also a head of the structure, membrane structure of membrane proteins. Professor Arkin did his studies in the Hebrew University and Tel Aviv University. He did his PhD at Yale University, then was a lecturer at Cambridge, and after all this he came back to the Hebrew University. And as I said before, he is the head of the group that deals with the structure of membrane biology of membrane proteins. I don't know how much you know about membrane proteins, but membrane proteins are a tough thing to research. And in order to elucidate the structure and the structure and function relationship in membrane proteins, one needs to use special means. And what Professor Arkin will tell us today is a little bit about the theory and experimental tools that are being used in order to research and understand the function and structure of these proteins. He's dealing with computational methods as well as experimental methods, part of which they have developed in their own laboratory. So, Professor Arkin.
Toda rabba. Okay. Hello, everyone, and welcome. Can you hear me in the last row? If you can, please wave your hand. Okay. Are you the only one, or other people are tired? Okay. So, what I want to talk about today, or to expose you to two things, I guess. So you're getting two things for the same price, for the price of one. And that is the topic of membrane transport and also computer simulation of biological systems. And when I talk about membrane transport, a lot of people sort of shy away from it because it's not as sexy as other things in biology today. People are really excited about genes and systems biology, and we are in the post-genomic era, etc. analysis, and membrane transport seems sort of very petty. You're only dealing with a very simple thing, taking something from one side of the membrane and moving it to the other, and it looks rather boring, right? And in fact, membrane transport, or the one that I will be discussing today, talks about this. Okay, that sounds very boring. Salt. Of all of the interesting things that we find in living organisms, proteins, DNA, sugar, lipids, salt might be one of the most dull things that you can imagine. Sodium chloride, exactly the same thing, or potassium chloride, exactly the same thing that you find over here. Okay? And why am I talking about this? Because we all know of the phenomena of diffusion. Okay? If we have, as we have over here, we have a permeable membrane. Permeable membrane, I mean a membrane in which the solutes over here in orange can transport through that. We know that if at the beginning the concentration of the solute is not even, if we will wait a long period of time, what will happen? It will equalize. We will find the same number of particles on this side as we would on that side. Okay? Interesting considerations come when those solutes are charged, right? Look what happens over here. Here, if you count the number of circles, you see the same number on this side as you see on that side. But you also see that there's more positive circles with a plus on this side, and here there's more negative circles. So of course what you will have over here is diffusion that is not only triggered by concentration, but also by electrostatics. Okay? So this is what we call membrane transport. Okay? The two things that we need to remember are the concentration of the solutes and their charge. Now, what would happen, or where does the interesting part start taking place, is when this membrane over here is impermeable to the solutes, or to some of the solutes. What does impermeable mean? It means, for example, that the red circles cannot go through, but the black circles can. Then you will immediately realize that this situation will not take place, will not arise. Why? Because the positive charges from here, or the positive charges over here, will not be able to move. Okay? So the control of transport through this membrane is of critical importance. Okay, so remember, we're only dealing about salt, but why is it of critical importance? Because of this. Okay, this is not what you will be getting for lunch today, although you might want to. But this is here to represent food. Okay, and energy. Can anyone predict what is the relative amount of energy that we use, percentage-wise, in order to move ions from one side of the membrane in our bodies to another? Okay? So right now I'm moving, I'm walking. So you would say I'm expending energy in the muscles. Okay? When I'm running, I'm expending even more. What percentage of the energy intake in our body do we apply to membrane transport? Does anybody want to bet? Okay. 70, 70, 17? 70, okay. Do people think it's large or small? Large, okay. So it can either be as low as 0% or as high as 100%, okay. The answer is actually you're not far from the truth. It's about 75%. So when we think about energy utilization, so the reason that you're eating this hamburger is not so that you need the energy in order to replicate DNA, or to translate proteins, or to manufacture sugars, or to synthesize lipids. 3/4 of the energy in this beautiful cheeseburger goes to moving ions from one side of the membrane to the other. Okay? And mostly, I mean taking potassium and putting it inside the cells, and taking sodium and moving it outside of the cells. That tiny little difference between sodium and potassium is responsible for 75% of our energy intake, okay? So this is very important. Now, another thing that's important in membrane transport is this slide. Far less colorful than the beautiful hamburger that we saw a second ago, okay? Bioterrorism. One of the biggest aspects of bioterrorism that we either find unnaturally or naturally in these sorts of organisms is interference with membrane transport. Okay, so who knows what this fish is called? Blowfish. Blowfish. What's another name from the Far East? Fugu. Okay, why is fugu fish dangerous to us? Why do we need a specially trained chef in order to clean the fugu fish so we can eat it? What is the toxic compound in the fugu fish? Pardon? Nerve poison. What is the actual name? The actual name is tetrodotoxin. Exactly. Okay. What does tetrodotoxin do? It blocks sodium channels. Okay. So you can immediately see all of these creatures, by the way, exert their toxic effect, their lethality, by blocking channels, by blocking transporters. So you can immediately see that all of these creatures have utilized, again, conotoxins, scorpions, venomous frogs, red algae, fugu fish, etc. do this by interfering with your transporters. Now you might think, wait a minute, these are all really, really strange creatures, okay? Here's some more. Okay, some more creatures, and you can see the plant. Bees, for example, the biggest component in bee venom is a tiny little protein called melatonin. Melatonin doesn't block your transporter, but what does it do? It adds another one. So one way to interfere with transport is to add another transporter that screws with your transporter, okay? So again, the toxicity of all these creatures, including cobra venom, etc., is to interfere with with membrane transport. And another thing to realize that also these creatures do exactly the same way. And I don't mean those with 4 legs. I mean the guys over here. We also harm bacteria by ruining their membrane transport. Okay? That is how our complement system, one of the major components in the immune system, ruins or kills bacteria by inserting a huge channel that destroys all transport activities in bacteria. Okay? So how do we study this very important phenomena? Well, we do this also by experiments. So this is a scheme that is meant to show you sort of an experimental lab. But since you are young fledgling students, I'll ask you, where is this scheme taken from? Does anybody recognize what this is?
The experimental trying to build nucleotides.
Exactly. This is the famous experiment by, does anybody remember whom? So this is homework that I'm not going to check. Okay, so Google this. This is the experiment in which people took water. Okay, Julian Miller. Okay, I gave it up. They took water in some very basic compounds that you find in outer space. They elevated the temperature. They added electrodes, all this sort of stuff, and they actually got amino acids and nucleotides. So this is how life was created. So this is a way of experiments. And we also do simulations, okay? Computerized experiments, okay? Now, have any of you ever seen the movie The Graduate with Dustin Hoffman? No one ever saw the movie The Graduate? Okay, late 1960s, one of the best movies of all times. I really suggest or recommend that you see the movie. But one of the very famous scenes in the movie is that this recent graduate from university, which you will become in another 2-3 years, is talking to someone, and the someone is telling him, look, here is my advice to you. Go to plastics. Everybody will be working in plastics. So here's my advice to you, okay? Simulations, molecular dynamic simulations. Okay? I'm going to show you the power of that, especially in the field of membrane transport. And it is my biased view that in another 10 years or so, you'll find far more people doing molecular dynamic simulations than you find today. And you will find far less people doing experimental biology that you do today. In my mind, fields such as experimental structural biology might disappear altogether. Okay? And I'll give you a few examples why. So, what is the basis of this brand new field of molecular dynamic simulations? To simulate a molecule on the computer. The basics is 17th century physics. And I very much hope you know who this guy is. This is the time when you actually say his name out loud so that I can recognize that you do instead of going like this. This is Newton, okay? And all the basis of molecular dynamics simulation, again, is 17th century physics. It's all Newtonian physics. So it's not that I'm saying that you should forget what you studied or what you heard about 15 minutes ago about quantum physics, but in molecular dynamics simulations, all that we're trying to do is to solve Newton's law. And the idea is very, very, very simple. The idea is, I know what the masses of the atoms are, right? What's the mass of carbon? 12. What's the mass of oxygen? 16. What's the mass of hydrogen? 1. Very simple. I only need to look at the periodic table of elements. Then, for me to know where that atom will move, all I need to do is to figure out the forces on that atom, right? Because that's what Newton told us. The force is equal to the derivative of the momentum. F equals ma, right? This is the mass. I know what that is. And all I need to do in order to figure out where is this atom moving on my computer is to figure out what is the force that's acting on that atom. Okay? Really, really, really simple stuff. Okay? How do I know what the force is? Once again, we go back to Newton. So what is the force that is acting on something? Force is simply the derivative of the potential energy. If you know what the energy of a particular atom is, you know what the force is. Okay, and I'll give you a very simple example. If the energy landscape looks like this, okay, and my atom is over here, what is the derivative of this function? 0. There is no force on that atom. It's not moving anywhere. Okay, but if my atom is over here, what is the derivative? The derivative is this line over here. It wants to push the atom down here. It's exactly the same sort of idea that if we have a ball that is moving like that, okay? Really, really, really simple stuff. The only slight problem is that in biological systems we have lots and lots and lots of atoms, so it takes a long time to calculate. But that's okay, that's why we have computers. And the computers nowadays are very, very, very powerful, okay? So how do we calculate the energy of the atom? Okay, so I'm going to go through this just so that you'll be exposed to this and realize it's simple stuff. The simplest thing is we calculate the bond distance between every two atoms. This is the first atom, that's the second one. And then we say, if the distance is too far away, the energy isn't good, it's too high. It should pull them inside. But if the atoms are too close together, once again, the energy is too high. It wants to push them away. Okay? Exactly like taking two balls and putting them on a spring, and then they will move like that until they find the optimal distance. Okay? So this is the optimal distance between them. This is too close, and that's too far away. And the equation is ridiculously simple. This is Hooke potential. Okay? We also do the same thing for bonds. Okay, so if this is the ideal bond angle, this is too small, and this is too big. Okay, once again, a very simple harmonic potential. Okay, and the only thing we need to do is to look at all the atoms and to figure out what is the current distance or angle between them, and then knowing what the optimal is, applying that energy. Okay? There's also energy for dihedral angles and also for improper, etc. Okay? Now, all of this stuff is very easy. Why is this relatively easy? Because an atom is it can only be bound to maximum 4 things, right? If you're looking at methane, what's the formula for methane? CH4. So the carbon in the middle is bound to 4 hydrogens, max. Okay? Okay. The hydrogen is only bound to 1 atom, carbon. So calculating the number of interactions of that carbon is not very much. Maximum, let's say 5, 6, 7, can't be more than that. But every atom also has a charge. And then we need to calculate the electrostatic interaction between every one of the carbons and every other atom in the system. So if my system has 200 atoms, that's 200 square. If my system has 2,000 atoms, it's 2,000 square. So the electrostatic interactions, again, and this is a very, very simple law. This is Coulomb. Okay, Coulomb's law. Again, this is in 16th century. This is, I think, 18th century, but very, very simple. Okay? So this is what we call non-bonded interactions. These are interactions between the atom and things that are not bound to it. And I won't go through the van der Waals interactions as well. Okay, so what do we apply this to? I'm going to show you of a simple membrane transport system that we are looking at, that we want to understand how it works, and it comes from the influenza virus. Okay? So what you see here is what led to the 2009 recent influenza scare that was called the swine flu. Swine flu. It actually came out of Mexico. Okay, and what happened in the swine flu is that several different influenza strains lived inside pigs, mixed, and then something completely different come out. And this is what this movie is meant to represent. Okay, here you have the swine respiratory tract, the lungs, and you see a swine avian strain and a human strain. They're going inside, and together they're combining, and a brand new strain comes out, okay, in a few seconds. And that is a release of a mutated strain, and that was the swine flu that we were not used to. So this is what the influenza virus sort of looks like to the artist. And what you see over here is that the virus, this is the viral genome. The viral genome here is actually not composed of DNA, but it's composed of RNA. Many viruses have RNA instead of DNA. The viruses also have lots of proteins here on the outside. By the way, these are the proteins that our immune system recognized. There's two famous proteins, one called hemagglutinin and the other neuraminidase. Forget about the names, just remember H and N. And do you remember what was the other name of the swine flu? H1N1. It means that it was type 1 of protein called starting with H and type 1 of a protein starting with N. But there's also another protein here that's the one that we are interested in, this tiny little purple protein. This purple protein is a transport protein. Okay? And how does this transport protein work? Why is it important? So this is the virus life cycle. Okay? Now, just by saying that, I said something incorrect. Why can I not say the virus life cycle? They're not alive. Viruses are not living organisms, right? So there is no such thing as the virus life cycle. I can say the virus infectivity cycle. Okay, so what does the cycle looks like? So this is the virus on the outside. This thing over here is a cell. One of our cells in our body. The virus attaches itself to special chemicals on the outside of the cell. And then what it does is it fools the cell to think that it's food. What happens after that is that the virus gets internalized. You can see over here, the virus is inside the cell. And then what the virus does is it fuses its membrane with this membrane, this vesicle membrane. You see it's now fused, and the viral RNA is now inside our body, and infection starts. That's part of the cycle. How do we make new viruses? So here you generate more RNA. They go to the membrane, and here, amazingly, something opposite takes place. Here, instead of fusion, here budding takes place. Again, I will try to demonstrate. Here, the virus goes like this, opens, and the genome comes out. But here, the virus closes, trapping the DNA, the RNA, and goes away. Now you can simply ask yourself, how can this be? Because these two processes are completely opposite one another. Here, the virus is doing this, and here, the virus is doing that. How can we have two processes which are exactly opposite one another take place in the same cell? The answer is very, very simple, pH. This process is favored in neutral pH. This process is favored in low pH. That tiny little difference, concentration of protons, will shift the equilibria between these two processes. Okay? So how do I know that that transport system is important? I know because these two drugs, one is called Symmetril and the other is called FluMedine. Okay? These two drugs block that tiny little transporter. And these two drugs are also very good, very potent anti-flu agents. Now, I'm willing to bet a small amount of money that none of you have ever heard about this. And I'm also willing to bet now a large amount of money that many of you had the flu, right? Probably every one of you had the flu. So why didn't your doctor tell you about Symmetril or FluMedine? The reason is that the virus develops mutations very quickly against these drugs, making them completely useless. So when initially you use the drug, it's very, very good. It blocks the virus infection like that. But very quickly afterwards, a new strain of influenza comes that doesn't care about this drug at all. And that's what we're interested in. Okay? So let's see. So this is what the channel looks like. This is what this little transporter looks like. And this is what Symmetril looks like. And we think that Symmetril binds over here. And you can see Symmetril is now a plug. A very, very nice plug that blocks this channel. Okay? When Symmetril sits over here, this protein cannot allow acid to go through. And I have already shown you that the acid concentration, pH is another name for that, is critical for the viral infectivity cycle. Okay? Now the amazing thing is that this is structures of proteins from viruses that are resistant to the channel. Okay? So this is the wild type, okay, which we normally abbreviate to WT, I'm sorry. And you can see that the drug fits very nicely here. But the virus can undertake two different mutations. This means that amino acid number 31, which used to be S or serine, is now N or asparagine. Okay? This mutation renders the virus resistant to this drug. This drug is useless. You can see that the binding pocket now is much too small and the drug simply doesn't have a place to fit. Okay? But amazingly, here's another class of mutations. Here, this we change amino acid number 27 from V, valine, to G, glycine. And what this did is actually made a larger binding pocket. Okay, so what does the virus do? Again, this is a hole that the blocker, the drug needs to fit inside to block transport through this hole. The virus has two ways of avoiding this drug. Number one, what it does, it makes the hole smaller. So your plug can't fit inside, right? If it can't fit inside, it won't work. Okay? But another way that the virus overcomes is it actually makes the hole bigger. What can happen then? The drug can fit inside, but it doesn't form a tight seal. So here, transport still takes place. Okay? So this sort of molecular picture, which we arrived at using simulations, shows us how this drug works in the case of the wild-type virus, and two completely different ways that the virus avoids this drug by generating mutations. Okay? So that's one example. And this is the sort of simulation that we run. And you can see, what do we have here? This red stuff over here, what is this red stuff? Anybody has any idea? What are these red and white balls? Pardon? Water. Exactly. Red is oxygen, white is hydrogen. Lots and lots of different balls. Okay, what are these things with a blue? Phospholipids, lipids. They are the major component of our membranes. Okay, and what is this thing in the middle in sort of a golden color? That's the protein. So what we do is we simulate all of this together, and then we try to see, do things actually go through the membrane? Okay? Note that what we're doing here is we're only simulating actually one protein. Okay? The protein is composed of 4 components, and there's only one of them here. Okay? We want to simulate this long enough to make sure that what we see is actually correct experimentally. Okay? What we were also able to show is how this protein is activated when you lower the pH. So this protein, now it's going to get slightly complicated, not only transports acid, but is also triggered by acid. So the protein opens when the acid concentration increases, and then it allows acid to go through. Okay? So what we were able to show is that upon increase in concentration of acid, the protein undergoes this sort of rotational change that you can see depending on the concentration. Here at low pH it's open, now slowly it's going to close. Okay? And again, this is the sort of thing that you can do with molecular dynamics simulations. Okay? By the way, none of you have had any questions up until now. Feel free to interrupt me with any easy questions. I'll be more than happy to take them. Hard questions you can ask your friends. So, we then wanted to ask a more basic question. How does this channel blocker work? Okay, again, transport is really important. Transport takes place through a hole. What does it mean that this anti-flu agent, Symmetry, blocks the channel? Is it simply like this plug over here? Is it a physical block? Okay? So what we did here is we actually ran simulations and we calculated the electrostatic potential. And I'm going to go through this very briefly. This is what it looks like. Okay? So in red, we see areas of negative potential. What does a negative potential mean? It means that if you have things that are negatively charged, they won't want to be here. They will be repelled. But if you have things that are positively charged, they would like to come here. Okay? Because they get attracted. So what we see here is that this is one side of the membrane. This is the other side of the membrane. The lipids are pretty much neutral. They don't have any charge. Okay? And what you see over here, this is the protein. If you are an acid, a proton, you're positively charged, you'll be very happy to move from here to here. Why? Because you're welcome over here, because this is negatively charged, and you're welcome over here because it's negatively charged. And you're not going through any area that will repel you. No blue area exists between here and here. There's a bit over here and there's a bit over here, but there's a nice road for you throughout. What happens when we add the drug? When we add the drug, you can see that that's no longer the case. Okay? When we add the drug, you can see that if you are a positively charged species over here, you can't go here because you're going to encounter this positive electrostatic barrier. So that means that this plug that I alluded to a while back is not really a physical plug. Okay? What I showed you over here is not a real physical plug, but rather it's an electrostatic plug. So the way that tetrodotoxin works in the fugu fish, okay, we don't really know because the structure isn't solved. But in this case, we have direct proof that the way that the plug works is not by physical barrier, but by electrostatic barrier. Okay? Now I want to show you, what else do I want? It has to enter the channel, correct, yeah. Okay. Well, there isn't a physical element because when you bring a channel blocker that is exactly the same size but is uncharged, it doesn't block the channel. Okay? So I'm going to show you a schematic of how this protein works. Okay? So what is this protein? Again, this is a transporter from influenza. This is 2 parts of the protein. There's actually 4 of them symmetric, but doesn't really matter, so I'm sparing you those. And these are 2 important residues. This is residue number 27, and this is residue number 31. Okay? This is the wild type protein. In the wild type protein, what happens is that there's a really nice space over here. We call this sort of space a binding pocket. There's a very nice binding pocket for this drug over here. Here I didn't call it by its generic name Symmetril, but rather its sort of chemical name, which is amantadine. Okay? So you see, there's a really nice space for it. And the drug, because this is positively charged, it exerts a positive electrostatic potential. That's this blue halo over here. So this is how the drug works. Okay? How does the virus develop resistance? Here's mode 1. So the first way, the first type of resistance, look what the virus did. This residue used to be small. It used to be S. Now it's much bigger. It's N. These residues are bigger. It means that the drug can't fit. There is no room for the drug to fit. If the drug doesn't fit, it means it can't block and protons can go through. Okay? So that's mechanism resistance number 1. Another mechanism of resistance is this one. Okay? This is mechanism of resistance number 2. What happened here? Instead of this residue becoming bigger, this residue became smaller. That means that the drug fits, but it's moving all over the place. Because the drug is moving all over the place, the binding pocket now is much bigger. What's going to happen? No positive electrostatic potential, so protons can go through. Now, without this knowledge, we will not be able to design a new class of anti-influenza agents. Okay? So, I'm slightly running out of time, so I'm going to skip this part, and I'm going to go to a really interesting part, and that is specificity. So when we look at transporters, I told you the basic reason that our nervous system works is that we have transporters that can tell the difference between 2 very, 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 and I can add some more varies after that, similar ions, sodium and potassium. Okay? They can tell the difference between sodium and potassium. A sodium channel and a potassium channel are each specific for sodium and for potassium. Potassium channels in our body transport 100 million ions per second. And still, despite the fact that they're doing this 100 million times per second, they can tell the difference between potassium and sodium. They are selective. And the question is, how does this work? Because again, what's the difference between sodium and potassium? Do you know what the difference is? 0.3 angstroms in diameter. Okay? So one is a ball this size, the other is a ball slightly smaller. And the difference is 0.3 angstroms. An angstrom, to remind you, is 10 to the minus 10 meters. So this is 0.03 nanometers. That's the difference. How can it do this 100 million times a second? Okay? Now, the ability to distinguish that is what allows all neural conductivity. Okay? Neural impulses mean flux of ions. The only reason that we can get flux of ions is because we can distinguish between sodium and potassium. So this is critically important. Now, how do you generate that specificity? We were able to show that in one particular protein, the specificity is actually really strange. The specificity is not at the stage of binding. The protein binds both sodium and potassium. But in the case of the protein that we are studying here, it will only transport sodium. So why does it still bind potassium if it's not intending to transport it? Well, the answer is that it binds the two ions differently. It binds sodium with water all around it. But it binds potassium without any water. It binds naked potassium. Okay? So this is an example that shows you that in order to distinguish between what seems to us two very similar things, nature makes use of their inherent physical-chemical properties, and that is hydration. Sodium in water normally has 4 water molecules around it, maximum 6. Potassium has 8. It prefers 8. Now if you're asking yourself, wait a minute, I don't want to tell the difference between 2 balls. One is different from the other between 0.3 angstroms. I am going to tell the difference between a ball that has 8 water molecules or a ball that has 6 water molecules. That I can already tell the difference, right? That, you know, I can easily check how many water molecules do you have, okay? So the way that we actually go ahead and selectively discriminate these two similar ionic species is not based on the ionic species itself, but the ionic species and its water hydration level. Okay? That's a very important lesson. So the last example I want to give you is of a really big protein. And this protein is the protein that is responsible for the nervous system in our body. This is an amazing protein. This is a voltage-gated channel. So what does a voltage-gated channel mean? Channel is simple. It's a hole through the membrane that allows ions to go through. What does it mean that it's voltage-gated? It means that when a particular voltage is on the membrane, it's going to be closed, and when that voltage changes, it's going to open. Okay? It's exactly like sort of a switch that we have over there. When we press the electrical switch, the light goes on. We press it again, light goes off. Okay? So why is this protein the central component of all the nervous system? How does the nerve impulse move throughout our axons? Okay? The way that it moves is that, let's assume that the voltage started someplace. Okay? Okay, here I have a nervous impulse. The voltage in the membrane changes. It opens a channel. When the channel opens, ions go through. And what happens when ion goes through? It changes the voltage again. Okay? So then it impacts further channels. Because there's another thing that this protein knows how to do, and that is it shuts down after a given amount of time. So when you triggered it by changing the voltage, it will open, but then naturally it shuts down. So we can see that a nerve impulse moves through your system. So the question is, how does this protein work? How can you change the voltage and have the channel open or close? So this is a tremendous problem for the experimental structural biologist. How does a structural biologist solve the structure of a protein? How do we know what the structure of this protein is? Normally we take this protein and we crystallize it. We make crystals of this protein. We take these crystals, we put them in an X-ray beam. The X-ray beam diffracts. We look at the diffraction pattern, and from that, using mathematical ways, Fourier transform, to be specific, we figure out what the structure of the protein is. Okay, but then I want to do something simple. What does the structure of the protein look like when the voltage is different? I can't change the voltage in a crystal. Because a voltage difference means that the voltage on that the potential on this side is different from this side. And the crystal is an isotropic system. There's water all around. So unless I have a membrane, I can't change the voltage. So how will I know what does it look like when the voltage changes? So the structure that was solved was of course when the voltage was 0, right? It's isotropic. But the protein closes at minus 60 millivolts. By the way, you might think minus 60 millivolts, a voltage difference over here might not sound very impressive. 60 millivolts? What's a AA battery? The voltage? 1.5 volts, right? You know, the regular voltage that we put on our tongue is 9 volts, right? 50 millivolts sounds pathetic, but the distance here is only 30 angstroms, which is again 30 times 10 to the minus 10. So the voltage difference here is something like 10 million volts per centimeter. So it's a huge voltage difference, okay, because the distance is so small, okay? So how do we do this? What do we do? The answer is we run molecular dynamics simulations, and we simulate the voltage, because this is, once again, really easy. What does voltage mean? Voltage means that all you need to do is to multiply the force on each and every one of the atoms over here, and there's about 200,000 of them over here, and add to each and every one of them a force in the direction of the voltage. And then run this simulation for a very long period of time and see what happens. Now, what does a very long period of time mean? Okay, a very long period of time in this case means something like 100 microseconds. Again, you might say 100 microseconds doesn't sound very impressive. 100 microseconds, 0.1 millisecond. Not a very long period of time. Okay, so why am I still impressed by 100 microseconds? Because to run a 100 microsecond simulation requires us to do how many calculations do you think? Anybody have lots, okay? Can someone be slightly more specific? Okay, so let's work with this together. In order to do a simulation, I do a simulation in small steps. Getting from here to there is not by one leap, it's small steps. What's the size of every step? The answer is 1 femtosecond. What is femto? 10 to the minus 15, exactly. So if I want to reach a microsecond, which is 10 to the minus 6, how many calculations do I need to do? 1 billion, 10 to the 9th. Okay? Now you would say, wait a minute, that sounds foolish. Why don't you take bigger steps? Why don't you do bigger, longer simulation, longer steps? Don't take a step every femtosecond, take a step every nanosecond. That's not going to work. Do you know why? Because remember, we're simulating atoms. So again, I have two atoms, they're bound between one another. Right now, we started the simulation, the atoms are slightly too far apart. What's going to take place? They're going to move in this direction. If the step that I'm going to take is really, really long, what will happen? They're going to go like this. Okay? I need to take very small steps because I really want to simulate what takes place. So normal molecules vibrate naturally on the femtosecond time scale. Okay? How was this simulation done, by the way? Not with computers such as this or computers such as your laptops. This was actually done by a supercomputer. By a supercomputer, I don't mean taking lots of PCs that you can buy in Office Depot. It was actually done with a computer that had a CPU that was designed to do MD simulation and was built by a company called D.E. Shaw Research. And this is what the results look like, and this was published in Science last week. Okay, this is the protein. This is from the top and this is from the bottom. The hole is over here. Okay, you can see it shut down. This is what we call the voltage sensor domain. It has a red helix over here with lots of charges on it. You're going to see in a few seconds how this charge, actually it already showed that, let me go back. Notice how this voltage sensor domain is moving and then results in shutting this thing down, okay? I'm going to go like this again, okay? It's open from the top and from the bottom, and these guys, the red helices, are going to move, and in a second, it's going to close it, okay? You see? It moved and it shut it down. It's closed. Now this channel is closed. There is no way, at least that I know of, that experimentally you can come up with this answer, okay? Not as directly as this. The protein, by the way, is composed of a tetramer. Tetramer means that there's 4 identical copies. This is copy 1, 2, 3, and 4, okay? So there's 4 of these voltage sensor domains, and look at the time. Everything took place in around 100 some odd microseconds. If you want to see how this is done on an individual protein, this is one of the subunits. Look at what's going to take place on this red helix over here. The channel goes, by the way, is through here. I'm only showing you one of the units. In a few moments, this red helix is going to move. And this small difference over here, it's going to move this yellow helix. It's going to close the protein. Okay, going to shut this channel. Going to close it. Wait for it. Wait for it. Here it's coming. That's it. Okay, now you might say, that's it. This movement is what allows all neural activity in your body to take place. Okay? Again, this contains charges on it, 4 charges, 4 positively charged residues, arginine to be specific. They respond to the voltage difference, and then they physically move, and they change, they drag this helix, and they shut the channel down. Okay? How do we know all this? If you mutate the residues, if you remove the charged residues on the red helix, it doesn't work again. Okay? So I think with this I will pretty much stop, 58 minutes and counting, and I'll be more than happy to sort of accept any questions. Again, this was just here to give you a taste of what membrane transport is and then what computer simulation of membrane transport can do. No questions. One question. Yes. Everything is composed of atoms. Okay, so what we do is you start the system, and this is the trick. How do we know where to start? Let's assume that we know what the structure is in the beginning. Someone told us. Then what we do is we calculate the energy of each and every one of the atoms. The atoms in the water, the atoms in the protein, the atoms in the lipid, everything. And then we move all the atoms by a small amount, because that's how they would move in 1 femtosecond. Then we stop, and you know what we do? Everything again. We calculate all the energies, we move them a small amount, stop, calculate all the energies, and we do this 1 billion times.
And always it turns out the same way?
No, it shouldn't.
So how can you tell this?
Why, by the way, should it not always become the same way? Chaos. Okay? There's random fluctuations. The other name, by the way, for random fluctuation is temperature. Right? There's Brownian motion. So what you do is you repeat the simulation several times, and that you see that regardless of the random fluctuations, you reach the same answer. But even if when you repeat an experiment, you don't always get the same result. You shouldn't, because there must be a random component. I mean, Boltzmann told us that, right?
Is there any non-computer proof that we can apply afterwards?
Absolutely. So what you can do, for example, here, is when you get this molecular picture, you can see, wait a minute, I didn't know that this helix is moving. Then you can basically see, wait a minute, is this helix, are these residues conserved in evolution? If they are, okay, it means that they're doing something important. If I mutate them, will bad things happen? If I mutate them, will I be able to change the activity of this channel? What you need to do is to check using, does this explain does this explain experimental data that we know of? Okay?
You said that ion transfer took a lot of energy. Are there any research in this field that search for more efficient?
Well, okay, so I'll repeat the question for those you didn't hear. The question was that ion transport takes a lot of energy. Is there any research to try to overcome that, to find more efficient ways? The fact that it takes a lot of energy is inherent. It's not because these machines aren't very good. It's simply, if you look at a Boltzmann equation, delta G equals minus RT, the natural logarithm of K. If you want to have 5 millimolar potassium on the outside, and 150 millimolar sodium on the outside, and on the inside, exactly the opposite, that's the energy that it takes. Okay, it's not that these machines are wasteful. The energy is inherent in the concentration difference. Now, I'm even going to say something crazier. There's a lot of research that is going into making the machines less energy efficient. So the food industry is working on proteins that poke holes in your membranes as diet pills. Because the moment that you insert a channel in your membrane that is working against your natural channel, it means that you can eat that wonderful hamburger and have the energy go to waste. The only problem is, what does energy go to waste mean? What does it mean? What does it mean that you dissipate energy? Where does it go to? Heat. So you're going to be very skinny, but you're going to be very hot. Okay? And you won't believe the huge amounts of money that are going into ruining your energy production. Or ruining is too strong of a word, making it less efficient. Okay? Yes, please.
So you mentioned that the pH triggered protein, which is changing its structure depending on the pH.
Correct.
So my question is, it can be channel-like voltagegated channel.
Well, gating of a channel means opening and closing. There's lots of different ways that you can gate a channel or a transporter. This is an example of a channel that is gated by voltage. The one in influenza is gated by pH. There's also other channels that are gated by the concentration of a small ligand, a small chemical, such as, famous examples are, okay, dopamine, okay, serotonin, all these sort of chemicals that we know that cause problems in our mind. Okay, glutamine, acetylcholine. So there's many ways that a channel can open and close. Voltage, specific chemicals, pH, and there's even other cooler ways. One of the coolest and nicest channels that we know of responds to movement. Where do we find these channels? In our ear. In our ear, we have channels that have tiny little hairs on them. And when sound waves come, the channels open. What does channel opening means? Means that the voltage in that area will change because ions will go through. What does that mean? That an electrical signal to our brain comes from a very particular region because a channel is now opened. Who can give me another example of opening a channel?
Under receptor.
Photoreceptors, light. Light is another one, okay? Actually not in our mind, in our eye it works in a different way, but in bacteria there's lots of channels that open by light, okay? Yes?
So you say that you think that simulations are the way of the future?
Absolutely.
Is that because you think that there is no possible way that physical or real world experiments can get the same results?
No, I wouldn't, well, I wouldn't say there's no possible way. I would say that when I look at answering a biological question, I always ask myself, what is the best, which is another way of saying the easiest tool to answer the question, okay? And if the easiest tool is to put the experiment on the computer and to simulate, then that's what people are going to use. In this case, it's not the easiest, it was the only one. But again, these are incredibly expensive computers, but so is a synchrotron. Okay? But in the future, because again, CPUs are becoming so much faster, in my mind, we are now at the transition point where in another decade or so, you're going to see far more of these taking place. I'm not saying that experimentalists will be out of a job. All I'm saying is they're going to be doing different kinds of experiments. Ah, sorry.
How long does it take to simulate such a simulation?
Between you hit enter until you get the results. Here, again, on the special supercomputer that there's only about 20 of those in the world, it took about 3-4 weeks. But that's nothing. What is 3-4 weeks? By the way, what is the biggest expense on these computers? What do you think? Is it buying the hardware? Nope. Electricity. The electricity bills on this computer is huge. And where do they actually put these computers? In power plants, because what they also need is cooling to cool the CPUs. Okay? So the actual, if someone were to give you this computer for free, you would go bankrupt. Okay? It's exactly the same sort of thing as someone comes and give you a Rolls Royce, you would go bankrupt because you wouldn't be able to afford the insurance.
Since we know so much about channel, can we like synthesize artificial channel
Well, we're always trying to synthesize sort of artificial channels, artificial channel blockers. So many of the sort of very dangerous creatures that I showed you, either fugufish or the cone shell or the cobra, we use some of those venoms for human health. One of the best examples is, you're relatively young, so you don't need that. What is the most famous and common cosmetic treatment? Botox. Botox. What is Botox? Botox is a small protein that comes from a bacteria called botulism. And it's a toxin. What does that toxin do? It destroys a protein in the neurons, and those proteins are there in order to generate membrane transport. Okay, they actually kill fusion of two vesicles. So here is an example of a toxin that interferes with membrane transport, but used in a very small amount is good for us, for those of us that want less wrinkles. Okay? But many of the drugs that we take for hypertension, if your heart is working too much or working too low, everything that they do is change membrane transport. Okay? Something like 30 to 40% of all the drugs that we take, that the doctor can give us, affect membrane transport. Okay? An amazing number.
You said that we could block the channels with artificial drugs. Is this also the case, and then the viruses develop resistance?
Absolutely.
Is this also the case for natural?
Okay. Okay. Very, very, very good question. Okay, so influenza has a channel, and we have a channel. We block the influenza channel with Symmetrel. The fugufish blocks our channel with tetrodotoxin. Why don't we develop resistance like influenza, right? That's basically the question. The answer is that influenza and we use completely different evolutionary strategies. The strategy of viruses and all microorganisms is to make a huge number of microorganisms, each slightly different from one another, and if one of them succeeds, they win. Okay? Our strategy is to make very, very complicated organisms and to make sure that it can fight the next stage. Okay, so it won't be to my evolutionary advantage if I die from this infection, but someone in the room survives. In bacteria, that is the case. Okay, so it's a different way to fight evolution. One is rapid change, and the other is building a very, 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 very sophisticated machine.
Arkin.